Strand Seq is a method for single-cell DNA template strand sequencing. It was originally developed for tracking DNA strand segregation during cell division, but has many other applications as well. During DNA replication, a complementary nascent strand of DNA is made from each original template strand. This creates two identical sister chromatids for each chromosome. During mitosis, sister chromatids are divided equally over two daughter cells, ensuring they inherit identical DNA complements. Because the DNA in both daughter cells is identical, it is impossible to see which cell inherited which template strands. However, if cells are pulsed with a dose of the thymidine analog BER-DU for exactly one cell division, the BER-DU becomes incorporated into the newly formed DNA strands, making it possible to distinguish the sister chromatids. Upon cell division, each daughter cell will inherit a template Watson strand which does not contain BER-DU, and one nascent Crick strand with BER-DU, while the other sister chromatid will be the exact mirror image. In order to detect inheritance of template DNA strand at the single-cell level, single cells are sorted by means of flow cytometry. The DNA is extracted from each cell and then fragmented. These DNA fragments are further processed for sequencing. The BER-DU labeled DNA is removed, leaving only the original template strand behind. Each DNA fragment is then amplified, sequenced, and aligned to the correct location in the genome. Further analysis is performed by a software package called BAIT, which plots the reads on ideograms and determines the strand state for each chromosome. A chromosome may inherit one Watson template strand and one Crick template strand, each from one of the homologs. It is also possible that a chromosome inherits two Watson or two Crick strands. So, a cell with 46 chromosomes will look something like this. Using Strand Seq, we can now determine which template strands were inherited for each chromosome. And by comparing strand inheritance across many cells, we are able to determine if it is random or not. Interestingly, not all chromosomes inherit intact template strands. Instead, they inherit part of the Watson template strand and part of the Crick template strand. Such a pattern indicates the presence of a sister chromatid exchange, or SCE. These are the result of a DNA double-strand break, and the number of SCEs is an indicator for genome instability. Another type of rearrangement is the chromosomal inversion, resulting from a broken chromosome that was put back together incorrectly. If two chromosomes break at the same time, pieces of the different chromosomes are sometimes pasted together wrongly. This results in chromosomal translocation. Strand Seq can also be used to detect chromosome copy number variations across the genome. It is also possible that chromosomes are not evenly divided over the daughter cells, resulting in a cell containing, for example, 3 copies of chromosome 18 trisomy. Strand Seq is a very powerful tool for detecting not only DNA inheritance patterns, but also different types of chromosomal aberrations. As such, Strand Seq can be used to assess genomic instability at single-cell level, resulting in a much higher sensitivity than previously possible.